Good evening and good morning, dear participants. This is Berin Lukas, Head, Department of Sociology and Industrial Relations, St. Joseph's College, Bengaluru. On behalf of our principal, Reverend Father Dr. Victor Lobo, staff and students, I extend to you a very warm welcome to this webinar, Search for a Cure Against COVID-19, Data Analytics, Virtual Screening and Drug Discovery. It is also my privilege to extend a very warm welcome to our distinguished speaker, Shri Chris Lewis. We have 736 registered participants. I thank each one of you for this overwhelming response. Welcome once again and thank you.
Good evening, participants and dear students. It's my privilege to introduce a dynamic speaker, Mr. Chris Lewis, for this evening International Webinar 3. Mr. Chris Lewis is the pioneer of computer education in Karnataka state. Is great IT expert, helped St. Joseph's College to establish computer science department in the year 1986 for both PUC and degree college. This is considered as the first computer science department in South India in college level. He is internationally known consultant for many decades. Mr. Chris Lewis has made large number of presentations at various institutes of higher learning. He was a director of Global Stock Trading System, NYC Management Consultant, Data Analytics, Sea Change International. And he was a product manager, business intelligence, ERP system, Bell Atlantic NYC. And he also a good friend of Sudhamurthy Infosys. From last 25 years, he is very much into global IT. So this is a small introduction of today's speaker, Mr. Chris Lewis. Over to you, sir.
Okay, thank you. So I can start my presentation?
Yes, sir, you can start.
Okay, thank you very much. Good morning, good evening, folks who are attending this webinar. So you can see the presentation?
Yes, sir.
Okay, I don't have to do anything, right? Okay. Okay, so this webinar is going to be... Yeah?
Carry on, Chris, it's clear.
Okay, so this is the following topic I'll be covering in this webinar. The main theme is drug discovery by utilizing bioinformatics and artificial intelligence. So I'll go over a little bit of viruses because the whole presentation is geared towards COVID-19 virus and its impact. There will be a short talk on COVID virus, then series of case studies on structural analysis, sequence analysis, and medical diagnosis, and data analytic and virtual screening. To start with, the bioinformatic field, we are using artificial intelligence to process its data. Especially in biology, there is a lot of questions they have when it comes to its genome and protein structure and internal activities. And artificial intelligence is a field where it has been considered as a fourth industrial revolution after the first being an electricity and internal combustion engines and telephone, and second is computer, and third one is internet technology, and this is a huge industrial revolution, fourth industrial revolution, where this artificial intelligence and its subset, which is going to literally change the world, as well as it will have impact in most of the sectors which is applied. Now, here the all areas where the artificial intelligence is applied on your right hand side, it has machine learning, speech synthesis, and computer vision, visualization, and natural language processing, and automation, and IoT, and virtual reality, and augmented reality. In this presentation, I will be covering the following case studies using machine learning. There will be synthesis. Another one is classification of blood content. Then when it comes to speech, I will not be anything doing in this. Computer vision, medical diagnosis, and visualization is all analytics. It's very, very important because there's a lot of creativity you have to show in visualization. And NLP is gene synthesis and DNA synthesis. And automation, of course, nothing. I think I have done this seminars in prior in the college, especially. I don't know whether I have done. I think the autonomous car and other stuff. Now, if you look at on the left-hand side, artificial intelligence, it covers a lot of areas, like predominantly a gene sequencing, in that immune therapy and gene editing. Immune therapy is basically, again, it's a bioinformatic field where you mobilize the immune system to take care of, it's a kind of boosting for immune system to take care of the immune system within the body, especially for cancer therapy. And gene editing is another field where you can edit the gene, insert, delete, and all those stuff. All those things you'll be seeing in the case studies, how to manipulate the genes. And basically, artificial intelligence is a deep learning and reinforcement learning seeks to mimic human ability of cognition to understand data, find patterns, make predictions, find recommendations without human interventions, or it's a self-learning. It's not like a old computing methodology. Here, we don't write any programs. You give data to the artificial intelligence machine learning system, and machine learning itself will, looking at the data, it comes up with the solution. As I present my case studies, you can see there is not much coding involved. Basically, you train the system by giving some labels to the data, or in some cases, clustering where you don't give the label. It will find out the pattern within the data and come up with the predictions and discovery and solutions. Now, in bioinformatics, biology part, basically the biological questions, data related questions will be answered using machine learning or artificial intelligence. So predominantly here we look into a DNA structure, RNA, then protein and molecular network, and cell, as well as physiology diseases. As here in DNA, gene finding and genomics and gene expression, as well as protein structure and protein structure simulation. So anything to do with the data, it is not a technology. You have to understand bioinformatics is not a technology. Only assist biology in virtual or synthetic way. You take a data and we come up with a prediction, right? Biotechnology depends on bioinformatics, biology depends on bioinformatics, bioengineering depends on bioinformatics, because the all processing of bio data, that is a metadata, is done by bioinformatic algorithms. The algorithms are used from artificial intelligence and machine learning. Now, at the informatic part, basically it's a data management. All the data of biology, that is gene-related data and other data, with a lot of data banks, and utilizing and modeling and algorithms of machine learning, deep learning, artificial intelligence, do a data mining for a biological discovery, and modeling, lot of modeling, because a lot of biological molecular interface, the intricacy of those interfaces requires a simulation or a modeling. Okay, so these following areas, machine learning is involved in solving bioinformatic problems, like a sequence analysis. Structured sequence analysis is for a DNA and RNA sequence, which we will see in COVID virus, how we are going to do. Structure analysis, that is the protein structure, that is every part of the COVID virus. Then expression analysis. Expression analysis is the genes can be expressed or not expressed, especially it's a lab on a small glass plate where the DNA spots are put, and the machine learning can classify a particular disease or not, whether it is a cancerous cells or not cancerous cells, and pathway. Pathway is mostly a simulation, metabolic pathways and regulations, right? Now, with that, the biology is a descriptive biology, and with machine learning, we'll use that data, raw data from a biological system metadata, convert into vector form in a machine learning, and come up with the predictive language. Okay, basically, an artificial intelligence machine learning is a newer technology, state-of-the-art, next-generation technology, which assists or solves a problem of a lot of biology. With this, we are assuming with the technology, assuming the drug discovery should be expedited. In the past, if you see all the vaccines and all, it took some 5-6 years. Especially till today, we don't have any vaccine for HIV/AIDS virus. Ebola took, I think, 5 years. But here, we are assuming within 2 years, we should have some sort of a vaccine or a viral treatment. Okay. Now, next going to be a brief introduction about viruses, because without this, pretty hard to understand the coronavirus. Now, as all of most of you know that virus is a small non-living parasite which cannot replicate outside of its own host. And there are two types of mainly viruses. One is called a DNA virus and RNA virus. Our focus is going to be RNA virus because the coronavirus is RNA virus. And though the RNA viruses are the polio virus, yellow fever virus, dengue virus, they all falls under this particular category RNA. DNA is a double strand, RNA is a single strand. And there is also RNA, DNA virus, mostly those are retroviruses. The viruses can be classified as in a structural form. There are 3 major structures for a virus. The COVID falls under this round structure, and this particular shape is retroviruses, and there are predominantly 3 shapes classification. And again, you can classify into the host where it infects. It infects everything, all living things, animal, plant, and as well as bacteria, right? Now, the important slide is here, the Baltimore classification of the viruses. The viruses can be classified into DNA and RNA of 7 classes, right, on the left-hand side. So they are DNA virus, RNA virus, and within that you have got this one HIV, that is called a retrovirus. Retrovirus is DNA as well as RNA. Now on right hand side we have got all the viruses which we are familiar with. If you take a class 1, it is an DNA virus, where all of you are familiar with chicken pox and smallpox and herpes virus, right? For these viruses we already have a vaccine, and this is a double-stranded DNA virus. Now class 4 is a common cold. It's called rhinovirus, because this common cold is not influenza. Influenza is a different than a common cold. It's a rhinovirus, common cold. It doesn't go below the upper respiratory, and everybody gets a common cold. And polio also belongs to that particular category. And this is a RNA virus. Within same group, we have got this corona, right? Corona, we have got SARS. That is the first which infected a human being, then MERS, and now COVID-3. Right? This is also RNA, falls under fourth category. Because they are RNA, single strand, they mutate very often. We'll see why they mutate. And the fourth is again an RNA virus, which you are familiar with, that H1N1 bird flu and swine flu. Okay. Then the remaining, those two, measles and mumps, we are all vaccinated for that. That is a MMR vaccine, measles, mumps, and rubella. And Ebola falls under the same category. That's also, I think they have a vaccine for that. And sixth one is HIV, and seventh one is this is RNA HIV, and seventh one is again a retrovirus, that is DNA. Okay. So how they infect RNA virus infect a human cell? Now, if you see here, human cell, this is the cell. Within the cell, we have got a nucleus and cytoplasm around. Now, RNA virus makes a entry inside the cytoplasm. It can't get into the nucleus, and it will utilize its machinery. The ribosome, it's a machinery. This is RNA strand, right? So using the ribosome machinery, it builds up its parts. Parts are all protein structure, which you will see what are these different type of protein structure. It builds its parts and it assembles itself, and then when there are enough viruses, it will break up the cell wall and spreads out. Okay, that's all its intention is. It's not anything else. It's just wanted to replicate, right? Just to show an analogy here, you can take this cell nucleus as where the DNA of a cell human genome is stored in chromosome, and it's a double-stranded. And if the cell wants to make any protein, the double strand has to break into a single strand, and it has to come out as the mRNA and feed into a ribosome, and that's how this is basically the factory or a kitchen of a cell outside the nucleus. And ribosome is the one which uses the RNA and builds the protein structure, right, for a virus to survive. And because it's RNA here, because that RNA is error prone, because there is no protection. DNA has a protection because it's a double strand. There is error checking, like in a computer science student, they know what is the error checking. There is no error checking here in the DNA strand. And in the process of building its machinery, these DNA can get corrupted, and when it gets corrupted, you will get a corrupted product output, and that is considered as a mutation. And when it gets mutation, some part of its it's not the structure. There will be a structural change. That's all. It's called a mutation. It's highly mutating, but it very rare in DNA viruses, which these two are RNA here, and the remaining are this is RNA, and these two are a DNA virus. The chances are very rare in DNA virus. Now, ultimately, whether it's a group 1 or group 7, group 1, 2, 3, 4, 7, the basic, all viruses, their job is to replicate themselves, right? And that basically utilizing any living organism, utilizing their cellular mechanism, build their protein structure and assemble and replicate, multiply. And this multiplication, this basically it is called a protein, and protein has multiple foldings. This is amino acid. This is the first level of protein structure. Second, the second level of protein structure, third and fourth, and it's a very complex folding. It's the structurally it's very complex. Now let us look at the SARS coronavirus. Okay, that's just a brief introduction. I don't want to spend more time on that. Now, in detail, we are going to look at the coronavirus structure here. Now, if you look at here, the coronavirus has an envelope, which is called a E. It is again a protein envelope. Then it has a membrane. This is a membrane. Then it has inside it's RNA, right? And it has a spike. Now the spike protein is called corona. That's utilizing the spike protein that virus makes entry into a human cell. And you can see here the RNA, how exactly packed inside the, it doesn't have a nucleus. Only thing is it has these envelopes, right? It is packed inside the, and this is a genetic material which it uses to replicate. Now, E and M protein, which is viral envelope, N protein is again, it's not the envelope is not shown because it's just an RNA, and there are total nearly 29,000 something odd. When I compute, it's little more than that, and 29 proteins are there. Okay, so that X, the spike which I have told you earlier, that is one which makes entry into the cell of 29 proteins. 4 make the structural. There are total 29 proteins, 4 structural protein, and remaining are non-structural protein. Now it binds to a receptor called ACE, that is angiotensin converting enzyme, which is in the human body, and this ACE2 receptors are found all over in the lungs, in throat, in kidney, in liver, in heart, in brain, almost every place. That's why it's so much infectious. Once it gets inside, it can bind into all these receptors and get into the cell.
Chris,
yes.
Chris, 1 minute, one
yes.
announcement. Okay.
Yeah.
All the participants who are not able to view the presentation, kindly go to the list of people. The presentation is broadcast. Is broadcast a separate person in the list. It should be visible right behind, right underneath the name of the resource person, that is Kursi Ruisus, and it should say presentation. So just go on it. You should see a pin icon, and please pin it in order to view the presentation. Thank you.
Can I proceed?
Yes,
Yes,
Chris,
sir.
please proceed.
Yeah. So, what is the difference between this and earlier corona? It is in studies they have found that it's binds much strongly than previous SARS-CoV, and also it utilizes human enzyme called furin to make an entry. Right. This is the findings what we have recently, but there might be more findings which are coming out from clinical studies. So the enzyme, the it has a special enzyme called a protease, and that is the one which helps the virus to make an entry into the human cell. Now, what I have just told you here, I'm just going to just let me close this. So what you are seeing here is in the simulation is a virus RNA, but the thing is I can't show you RNA because it's a double strand. That's how the structure comes. This is the one which it uses to as a molecular machine to replicate itself, because all the coding of its part is there in the actually it is a single-stranded RNA, and this is the one which injects into the cell, and using a ribosome it makes its part. And this is the envelope. So this is a whole protein structure. You can see how complex it is. This is again using that ribosome. It builds this protein structure as its envelope. And next is this one is a membrane protein. And the spike protein, which is of our interest, is a pretty large. I can't do a rotation. This spike protein is the one which is called a corona, which uses to make an entry into the system. And this is a solid structure of a spike protein. And the all research is going on this particular spike protein to find out how to inhibit or where to inhibit, and finding on the drugs. All those things are revolves around studying this spike protein structure. Takes a lot of memory. I'm just going to close this. Okay. So now, let us look at what we spoke all about as an bioinformatics and artificial intelligence. How exactly artificial intelligence and machine learning can solve this, help the bioinformatics to do a research and solve their problems. Okay, the methodology is like this. First, data collection, sequence analysis, structural analysis, then ligand and database preparation for molecular docking and molecular dynamics. Okay. Now I already have a case study here. I'm just going to explain, then I'm going to demonstrate the case study. The central dogma of biology is DNA in all living organism replicates itself. So DNA can transcribe. This is called transcription from DNA to RNA, because any protein building, that is a structural part of the human being, living organism, it has to transcribe. And also there is a reverse transcription, right? Reverse transcription, like retrovirus, HIV, they come with the RNA, then they do a reverse transcription, they construct their DNA. And RNA also can be replicated. RNA also replicates by itself. Then it gets translated into protein structure. That is, a ribosome is the one which does a translation. And this transcription is done inside the nucleus of the cell by RNA polymerase and other enzymes which are involved in this. Okay, this is the central dogma of biology. Okay, now I have DNA structure of all these viruses, which I'm going to show you. Now, what our the analysis is going to be: you transcribe the DNA into RNA, then translate into amino acids. From amino acids, you analyze. It's a very brief analysis. Actually, genomic analysis is very vast. What is required only, I have done some coding. And finally, at the end, once the analysis of a COVID DNA is done, using that, you compare that with the analytic. I'm not using machine learning at this time. You compare the virus genome with the other related viruses. right? Normally what they do is, how they found out is, there is something called blasting. You can just take this material and blast onto the internet, and it will find out closest matching. When you do a closest matching, you can see that 89% of the COVID-2 is related to SARS-CoV-1 virus, right? You can see in the graph also. Then 71% it matches with the MERS, okay, Middle Eastern. Then it has no much relationship with the, though it is RNA virus, Ebola virus. Then the MERS and SARS, their comparison is 69% between these two viruses. Okay. So after that, I'm going to do one more demonstration, because this is just a starting point, because it's a small DNA structure, easy to do analysis. The same thing you can extend using machine learning. You can actually do the human genome. You can take a sample of a human genome. You can run it through a machine learning program and compare that one into a, build a model, compare that into our closest relative chimpanzee and our friend dog, right? Now, if you see here, human genome, I have taken sample only 4,300, but human genome has 3 billion plus 3 billion, 6 billion base pair, right? Now, you have to consider that 6 billion as a population, because we are using a statistical model here, and I am taking a sample. I am taking a sample of human genome and building a model, right? Building a model, and if I get a good the percentage of accuracy, I can use that model to compare the human genome with the other species genome. That's also there in one of the case studies. I'm now jumping to, okay, what happened here? Oh, okay. To the program, all programs are written in Python. Okay, before that, just wanted to tell you here, what is transcribe. Now, this is I've taken a small sample of nucleotides, the genome. When I say transcribe, this is a DNA to RNA. DNA to RNA, everything remains same, only T becomes U, right? Uracil. If you see, only T becomes U. That is, from DNA, you're converting into RNA. So this is a whole analysis. I am taking a COVID-19 gene. Okay, you can see it's a pretty big. This is the whole genome of a COVID-19. You can see it's pretty big. Okay, that's why you need and it's a total nearly 29,000, nearly 30,000 nucleotides. Okay, human genome, I said 6 billion. Okay, it's very, compared to human genome, it's pretty simple. Now I run my transcribe. So you can see all T has converted into U. That means the original, it's a DNA. Now I'm converting into RNA structure. Okay. So from RNA, so I'm translating now. That is a transcribe, translating into amino acid sequences. You can see these are all amino acid sequences. Each star is, right, start, stop, right? So converting the whole genome into amino acid sequences. There are total, I think, in human 20 amino acids, right? Now this is a codon table for me to do a comparison of for amino acid. Then the protein chain. Okay, I'm looking at how many protein chains are there. Total functional proteins are 80 and total proteins are 775. Okay. So next is a protein analysis. So protein analysis has its own algorithm. You convert that protein analysis into a vector, a bag of words, because that's where I'm using that NLP, which I showed you, an artificial intelligence. Using NLP algorithms, you convert into bag of words, and analyze the protein content, then finally you do the comparison. This is where I do the comparison, COVID with the SARS, MERS, and Ebola, right? Run a comparison. SARS has total close to 30,000. COVID has again nearly 30,000. MERS has 30, and Ebola has some 18,000 nucleotides. So then GC content analysis, these people who know the GC content, they know, because I just know how to run the algorithm. And this series of analysis on the protein structure, then finally a comparison. Okay, now here I just do a pure comparison. This is just analysis, and you can see the scores here. 89% close related to SARS, because now this comparison is on a RNA level. You have to understand comparison RNA level, as well as the protein level. Okay, so this is the comparison. Then the 3D structure, which I have already shown that in the simulation. In Python program, I can also do the 3D structure. It's not pretty. 3D structure of COVID. Again, it's COVID is all done in the Python program. And this is SARS. You can see how complex. Basically, if you see, those are atoms. If you see a red, let me see whether I can zoom this. If you see, look, red, it is oxygen. Then this is black is carbon, and this is nitrogen, blue. Any protein structure will have a hydrocarbon, that is carbon, hydrogen, oxygen, nitrogen, and sulfur. If you take a carbohydrate, you have got just a carbohydrate, carbon, hydrogen, oxygen, and lipid also. Only those, the pair of, here also you can see it, let me see, zoom in, okay. So you can see all these atoms, atomic structure, you can see how complicated the spike protein is. Imagine to find antiviral drugs or even vaccine, how complicated it will be. Right, the same thing which I showed, but here it is done through program, through the program. Okay, so next quickly, yeah. So next one is, I am just going to show you this. Okay, human. Okay, so again, sequence is a human genome, small sample. Okay, take 3 genomes and build a machine learning model. I'm building somewhere at the down machine learning model with the sample data. So I get accuracy of 98%. This is, those are done a statistical model. They know what the accuracy, precision, recall, and this one F1 score. So once I have this closed model, then I can, this is a training, then I test against other genome. So if when I test against chimpanzee, I get 99.3%. Yes, we are close. Now, when it comes to dog, this prediction is not that good. Actually, I know, I went and checked online. It is actually, we are related to 84%, not 93. That means model is not doing that well when it comes to other genome, because it's pretty hard to build a model on a laptop. If you have built on a high performance computer, maybe I would have got a closest result. Okay, so next is healthcare, medical. So in medical, how the artificial intelligence is assisting healthcare hospitals by building these machines, like diagnosing X-rays and medical information using artificial intelligence, convolutional neural network, CNN, and multi-layer perceptron and machine learning. For X-ray, I'm going to show you that it is, if it is COVID, it comes as this, and with the other, your blood and other swab test and all those things, it will categorize into 3. And actual X-ray AI or a machine learning model is very complicated. It's in the picture it looks complicated, but that algorithm I can show it's not that complicated. And this is the case study. I take a patient data, which is a database, and read that database into my model, and then predict. The prediction you can see on the top, these are all COVID, and this is non-COVID. First I train the model. Once I train the model, then I give an unknown patient, and it will categorize whether it's a COVID or not COVID. That's just to assist the doctor, because it's in the ER. Doctors, every doctor can't go through the thing. Mostly the machine learning in the US, most of the hospitals have the machine learning model to analyze chest X-rays, and the accuracy of the model which I have is pretty good, 93%, and its detection rate is also pretty good for COVID. This confusion matrix, false positive, false negative, should have a 0 false negative. I got a 0 false negative here. This is the presentation. Okay, so and this is how that whole model works. And next one is blood sample. You take a blood sample, run again machine learning model, and this is the data of the patient. This is actual data, just sample I have shown. Then run the model and compare the blood sample with the PCR result, RT-PCR, reverse transcription, the polymerase chain reaction, which through the swab they get the data, and the results are very comparable. So the models. Okay. So X-ray. Okay. Let me just show you. Okay, I have data set. One is COVID infected and normal. Okay, now these are all X-ray images. This COVID infected, I got a huge set of data here. Right, this is all COVID infected X-ray, and another one is normal X-ray. Now, what happens here in model? You feed this information to a artificial intelligence deep learning model. Okay, you read the X-rays, then you run the model. So here you read the thing and you run the model, and then find out the accuracy. If the accuracy is pretty good, then you give this is the result of my model. You can see that if you see the number of lines are very minimal. This is the beauty of deep learning. You don't have to write many line of code. Only thing is you have to train the model. Okay, so these are all X-rays. Then once that model is ready, then you can give it to the hospital and they can run, and you can see the result here, right? And it's pretty accurate. You can see the COVID patient, and they are, I think it's more white means, I don't know how to read the X-ray, but the deep learning network is, they have got better eyes than human being. They can predict much better, okay? This is the one case study. And the next one, I'm a bit rushing, ma'am, because I don't have much time. Okay, so X-ray, okay, blood sample. Again, I've got another model. So this model takes a blood sample and compares with the PCR result, the patient information, and the accuracy also you can see here, the data. So this is the raw data. Okay, it takes time. Okay, this is the raw data of a patient. I don't understand, but if I feed to the machine learning knows how to categorize this data. right? That's the beauty of machine learning algorithms. Okay. So next demonstration is, okay, now this is straightforward. The analysis, before the analysis, this is for a statistical package. Oh. Okay, I think mission freezes, I'm not going to show that. It's basically a statistical package using IBM SPSS. I think it's there in the college. I've taken recent up to, I think, last week data and run the model of the COVID patient, the whole world data, but I have narrowed down only for India. You can see ANOVA, analysis of variance. I think for economics department, it will be very interesting. And also ARIMA. I think I'm not okay. You got here ARIMA model. It's a prediction model using time series. And next one is this is again another model. This is all Indian prediction. You can see Karnataka, the cases are coming down. I think this is the last week result. Cases are coming down. This is all state-wise. This is dropping down. This is a individual result, all states here, and also different countries comparison and the global view. And that model, quickly I'll so Python, okay, COVID. Okay, so this I think I have to run this. I'll run all because the graphs, it's running. Star means it's getting the data and it's running. Okay, as it runs, okay, I'll show you. Let it run. Okay, so this kind of analysis anyone can do, even in your college. You can have a general analysis. You can also encourage your student to do a social distancing analysis, impact analysis, SWOT analysis. There are so many analyses you can do, and the tools you can do a fantastic the visualization. Right, you can use either IBM SPSS or you can use a Python program. You can use R program or you can use Tableau. You can see the presentation, the visualization is stunning. Okay, it's a simple line of codes. This is just on a whole state-wise India. I have to just put the whole data. Data is the whole world data. Just put the particular country. It builds the chart for that country. And see, you can also do a Google map. You can zoom it and find out region-wise how many cases. And you can also compare India with the other countries. This is all the comparison: Wuhan, India, South Korea, and then you can do a predictive modeling. And see, this is global view. So I can zoom this. So I can see the global, how it changes, right? So this is all visualization, comes under the data analytics and artificial intelligence, machine learning. Okay, all visualization. Visualization is the key. You have to have a good visualization. Okay, so next one is this is a real-time prediction. Real-time prediction is it directly, if I run right now, it directly goes to the data set and gets the data. Real-time prediction is here. Real-time. So this is real-time prediction. I already ran, I think last week. I didn't get the time to run, and you can see at the end how it has done the real-time prediction, and the same thing which I have in my presentation. And you can see in Karnataka, this is Karnataka. I can do any country, any state, the same program. Only thing is in the beginning you tell which data, and you can see it's coming down. At some point it was negative somewhere in April. Okay. Don't have latest data, otherwise I would have, and these, some of the models, machine learning, it takes a long time. Maybe some of them takes a day, some of them half an hour. Especially that anything to do with X-ray and all, it takes more than 24 hours to run those models. So I already ran, and I'm just showing you. Okay. Now, next one is a drug discovery. Okay. I think I'm close, but before drug discovery, okay, I think I have not shown you one more, the comparison of open. Okay. So comparison of the protease, it is called 6LU7. You can look into the internet. Okay. COVID-19, HIV virus, MERS, and SARS and Ebola. Okay. Let us quickly see that. Okay, I'll close this one. Do you want to save? No. Okay. So this is Ebola, right? And this is HIV. This is a protein structure which infects the human cell. This is MERS. This is again a protease. We call protease is an enzyme which infects the, again, it's the corona structure of MERS. And this is SARS, right? And SARS, let us compare with the SARS COVID. Doesn't, okay, slightly different inhibitor. Okay. So as I said, look, if this, they're all made out of atoms. You have got oxygen, nitrogen, carbon, carbon chain it is. This doesn't look much complicated here. Then there might be somewhere some sulfur and nitrogen, right? Okay. So the last part is drug discovery. Now, drug discovery, okay. So what are the targets for drug discovery? Okay. So we know it is infects AC receptor, and that is a protease, which I showed you the structure. Now these are the areas like prevention of virus entering into the host cell, right? That is prevention of attachment and prevention of fusion. So that is the one target. Second target is prevention of viral replication and survival within the host. That is a viral protease inhibitor and viral nucleic acid and protein synthesis inhibitor. Now these are the areas where the researchers are targeting to find antiviral drugs. Vaccine is totally different world. It's a different approach. Not this is only for a antiviral medication, right? Now, in the emergency, we currently, I think FDA has repurposed some of the drugs which are being used for Ebola, that is remdesivir, and for HIV, ritonavir. And also they are using antiviral drugs too, some FDA approval, then HQ and HCQ, that is hydrochloroquine, which is used for malaria, that is a parasite. And mostly they are also using erythromycin for the bacterial infection, that is antibiotic. Okay, now So these are repurposed. Now the research is mainly on how to find a drug, how the discovery. There is a different ways, and information technology can definitely help to do a research and okay. So okay. So this is erythromycin. This is a chloroquine, hydrochloroquine. Then these are the drugs for used for HIV, and this is used for, this is a repurpose right now in different hospitals. They use FDA approved. Some of the FDA approved drugs, even in India, I think they are prescribing that. Okay, now in the discovery, so the methodology for a discovery is this. This is a virtual screening. What the artificial intelligence program does is, models does is, there are two approaches. One is structure based, like a protein. It takes a protein structure and find a ligand or a substance to dock that receptor of that protein, that protease which we have talked. Okay, the docking is, first you do a search using machine learning with the similarity search as well as the machine learning search. Then you narrow it down the molecules such that it can bind those receptors. Right now, the bioinformatics job is only to do the research and provide the information for a biotechnology or bioengineering. We are not building any drugs. Only we can do a virtual simulation, and this can be done from anywhere. See, most of the things which are done directly from the home, because all the data sets are available, and the machine learning which is used for these models. I'm just going to quickly show you the model. Okay. So COVID discovery. Okay. This is molecular similarity. You go into the database, you search for, okay. You take a molecular structure and do the library to the do their clinical such that which molecule they categorize this multi categorization which takes a least energy consumption. This is one of the technique. It's again used through using a machine learning, and another one is QSAR. Okay, I will just run the regression model. Also, you have a classification model. So again, Python program. So take the data, run it, verify the model, find out the accuracy, and finally do the recommendation of the molecules. We call the ligands, molecules or compound, or to the clinical laboratory, and then they will do the real test and see whether any particular ligand will bind to that receptor tightly. Some of them they might not. That's so. This is right now. These are the trials are going on throughout the world. Okay, so I think that's it. And it's a long process. Those who are, because I worked for a Merck way back for a couple of years ago. Merck and Co. was one of the clinical trial, a product called Zocor. To bring that one product, it took 20 years. You can understand how long it takes. Now with the artificial intelligence, machine learning, deep learning, we are presuming that time frame will be shorter. Because all these tools are available right now for biology to help the find the drug discovery, and hope within a year or two we'll have a antiviral as well as vaccine. With that, I'll conclude and I will hand it over to you, sir.
Thank you, Professor
Can I stop?
Chris Lewis. Thank you very much. Respected academicians and dear students, of course, many of you from the student community are from, I understand, computer science background. There are also humanities students here. Professor Chris Lewis, who is considered as the doyen of computer science education at the college level in Karnataka state, in today's talk has blended biology and information technology so well. With your catchy demonstration, you have made us understand, especially we academicians and many students from social science background, about the complexities involved, what's happening all over the world, what must be happening in the lab, etc. Now, sir, you have given us a lot of information. Sociology is a very, very accommodative social science. It's the mother of all social sciences. And though many of the things that you said are highly technical, but that's how sociology is also looking at various developments in the world. That's what sociology of cyberspace and medical sociology is looking at. So as a technical man, as a IT expert, your talk has nurtured interest for many of us in sociology of cyberspace and medical sociology. Thank you very much, sir. You spoke about the interface. You also discussed the possibility of social distance analysis, virtual simulation, machine learning, etc., which is likely to become a very important part of we academicians as we move towards online teaching in a big way in these days of COVID-19 pandemic. So on behalf of the principal of St. Joseph's College, Dr. Victor Lobo, head of the departments of Sociology and Industrial Relations, Dr. Berin Lukas, on behalf of our student association, Social Cest, the technical team, on behalf of my colleagues from both St. Joseph's Evening College and Day College, who are part of the organizing committee. It was great listening to you, sir. Though it was highly technical, many of us from the social sciences background have understood a lot of things because of your wonderful demonstrations, especially. The floor is now open for question answer session. The concerned teachers and the technical team head may read out the questions to Professor Chris Lewis. We request Professor Chris Lewis to spend some more time with us to answer some of the questions. I understand many of them are highly technical. So thank you very much once more, Professor Chris Lewis. Over to the open house question answer session.
Thank you, Anup sir. And I also like to thank Professor Chris Lewis for sharing valuable information on artificial intelligence and machine learning to our participants. So you're really taken our participants from technology to biology, biology to sociology. So thank you very much for that. So your first question, is there any possibility that the SARS-2 COVID-19 will not remain in this country after 2 or 3 months?
Will not remain in the country?
Yes, sir. Will not, shall I repeat the question, sir?
Yes, yeah.
Is there any possibility that the SARS-2 COVID-19 will not remain in this country after 2 or 3 months?
It's there is no way because if you look at the nature of the virus itself, unless you have a vaccine, everybody is vaccinated. That is a challenging thing, because it's not easy to bring a vaccine in the market. Because when you vaccine, you are vaccinating a healthy person, right? And the only one thing we can do is a social distancing. Other than that, there is no other way we can, in fact, you can say, chase away the virus. The virus is going to be part and parcel of our life, unless and until we have uh, antiviral drugs. Let us say, in case if you are infected, it can be cured, or vaccinated, or the best way for you is to build your immune system. There are a lot of people who are infected, they are survivor, right? Only thing is, when your immune system is not strong, the virus has an opportunity to penetrate up to your below the lungs and heart and everywhere, right? That's the only way. I think it will be a part and parcel of your life. I think you have to get used to it, and cautious, keep maintain the social distance, and all other rules and regulations which government are saying you.
Okay, sir. Thank you very much for that answer, sir. So your next question is, can you share your view about bacteria-based antiviral therapy for treating COVID-19?
Right now, means virus is right now they are using antiviral, but we don't know how much effective it is. Like anything to do with the bacteria, it is antibiotic, right? They are using azithromycin in almost all the hospital. We don't know whether it has any effect on virus at all. It's again a molecule. Um, Other than that, I have not seen, other than azithromycin, I have not seen any other drugs in the list they are using for COVID. Other than that one antiviral, is why they are using it's a broad spectrum. Broad spectrum means it can kill many type of bacteria. You have to understand, any drug, like whether it is a antibiotic or antiviral, they are molecules. Will that molecule have any effect on the virus, or can it disable its function? Right now we have not seen repurposed drugs other than azithromycin.
Okay, sir. Thank you very much. So shall I ask one more question?
Yeah, sure.
Yeah. So what is the difference between the different types of tests to detect COVID-19?
Right now we have got RT-PCR. RT means reverse transcriptase polymerase chain reaction, right? Reverse transcriptase. Why reverse transcriptase is, through the swab, they will take your RNA. RNA is very fragile because bacteria has an RNA. They extract RNA. It's a fragile. It's error prone. It has to convert it into DNA, right? Anything to RNA to DNA, I show you the dogma. It's called a reverse transcript, right? Reverse transcripted polymerase chain reaction. Polymerase chain reaction is, there is a huge procedure to that. It has to be heated, and you have to make a lot of copy. That means magnify. Magnify is not zooming like how we do in lens. You make a lot of copies of the RNA, the DNA copies of the virus, and you color code it. Okay, that is the one. Next is a chest X-ray, which I showed you the model, right? These are the only two things: chest X-ray, symptoms, and the polymerase test, PCR test. Then blood test will reveal whether you are in the past you are infected or not. Then if your blood test might show you some antibodies which are formed, that shouldn't be your emergency. Emergency is a chest X-ray, your symptoms, and the main test is only PCR test. Other than that, there is no other test right now available. For all viral this one only PCR test. And the guy who invented, he got a Nobel Prize for that.
Okay. Thank you, sir. Thank you for that answer. So next question. Why is it so difficult to create a vaccine for HIV and AIDS?
Okay, HIV is a retrovirus. When I say that, I showed you that the third, it's a structure is totally different. What HIV does is, now if you take a COVID, it enters into the cell, right? It uses a cell machinery. It is in the cytoplasm, because a cell, any living cell, human cell, has a nucleus where it keeps its genome. Genome is, you can say it's a recipe. Recipe is locked inside the nucleus, and cytoplasm has all the, you can think as an analogy as a kitchen, right? And the ribosome may be a chef. Okay, so it enters into the kitchen, and COVID says, look, I have my recipe. I want the chef to cook food for me. Whereas in HIV, okay, HIV virus gets into the cell and it will get into the nucleus because it has a needle-like a structure. It injects its DNA, not even RNA, and it injects its DNA into our genome structure. It will stitch, actually puts inside the genome structure, right? It's a very complicated virus. Till now, we have no, and it infects the T cells, right? So only thing is, right now, with the past, I don't know, 10, 20 years, we have only concoction of a lot of drugs, antiviral drugs to slow down its replication in the multiplication in the blood. There is no vaccine as such. Vaccine itself is a very complicated. If you get into bioinformatics, you will understand, because I myself was not aware of all this stuff. Only when I got into this field, I know how complicated even a machine learning to do a structural analysis of a protein, how complicated is the protein. When it comes to DNA, yes, we can do a fantastic job on a machine learning. The minute when you come to a protein structure, it's even as a machine learning with all these algorithms, it's one of the complicated. Even the docking, we try to do ligand docking on a protease, is you know, that we have to add the hydrogen atoms, you have to remove the water molecules, and there is an energy saving. You know, as a non-biologist, I myself finds, imagine, it's so difficult. Imagine people who are working, who do not have a computer access, where we are doing a modeling, where they are doing most of the biotechnology. They are in the lab, taking that burette pipette and adding all those stuff. They can't visualize the way we visualize in the computer model. At least we can visualize and see what exactly is happening, but that is not 100% proof. It has to go through a clinical trial. It has to go through an approval and all those stuff. The HIV is, I am not the right person to answer, but I know it is, knowing so far my knowledge in bioinformatics, how exactly it infects a human cell. I think it's a complicated. I don't know whether we'll have a vaccine or not. And making a vaccine also is not an easy task. Not only you can make quickly, maybe the distributing is a problem. How long it can be from the manufacturing site to the source site. A lot of things are there. You have to read up online. I don't have much knowledge about this vaccine thing. My short knowledge, where I worked for a Merck in a clinical trial, I know how difficult it is to make a drug. I not worked in any vaccine preparing projects.
Okay, thank you, sir. Thank you very much for that prompt answer. So over to Berin, ma'am.
Dave, do you have any questions? No? Okay, fine. Anup, can we wind up, please?
I have an instruction for all the participants.
Go ahead, sir. Neeraj, please go ahead.
Yes. So a feedback link will be sent to you in shortly. You need to fill the feedback link within 2 hours to be eligible for a certificate. Now again, there are again more mechanisms to see who has attended and who has not. So just because feedback link is not the only guarantee of receiving a certificate. Thank you.
Thank you, Professor Chris Lewis, for being our resource person for this highly technical session hosted by Department of Sociology, St. Joseph's College, Bengaluru. Thank you very much, Dr. Berin Lukas, who took the initiative to organize such a wonderful session. Thank you, my dear colleagues, Professor Maria and Professor Dept. Technical team assisted by Neeraj Savio and his wonderful team. Thank you, academicians from all over India and 6 other countries who took time off. Thank you, dear students from humanities, from natural sciences, and from computer sciences. Thank you all. Have a pleasant evening. The technical team may now give instruction for the others to leave, except the organizing committee. Please go ahead.